Chromosomes are important structures. While studying Mendel's experiment, we studied about the factors or genes. Example, capital Y, capital R, small r. These capital Y, capital R are genes. The genes carry the characters from one generation to another generation. These genes are carried by the chromosomes. Genes are arranged on chromosomes. So chromosomes are responsible for the transfer of characters from one generation to another generation. The term chromosome was coined by Waldeyer in 1888. Inside the cell, our body is made up of cells. Inside each cell, there is a nucleus. The nucleus has thread-like structures called as chromatin network. During the time of cell division, the chromatin thread becomes chromosomes. During cell division, the chromatin thread becomes chromosomes. At that time, the chromosomes appear as long thread-like structures. During cell division, the chromosomes appear as long thread-like structures. And this is a single chromosome. The chromosome has two identical structures, so left side and right side, called as chromatids. These chromatids are called as sister chromatids because they are identical. And the chromatids are connected by centromere. Centromere connects the two chromatids. Each chromatid is made up of spirally coiled thin structure called as chromonema. What is chromonema? Chromonema are spirally coiled structures. A chromosome is made up of chromonema. This is a chromosome. It has spirally coiled structure, that is chromonema. We can compare the chromonema like a chain made up of beads. A chain made up of beads can be compared with the chromonema. The chromonema has number of bead-like structures along its length called as chromomeres. The bead-like structures are called as chromomeres. Chromosome has spirally coiled structure called as chromonema. This chromonema is made up of number of bead-like structures called as chromomeres. So chromomeres are the components of chromonema. Chromosome has narrow zones called as constrictions. Constrictions are narrow zones of chromosomes. In this diagram we can find narrow zones. Here there is a constriction, this is another constriction, and this is also a constriction. One of the constrictions is called as primary constriction. The chromosome has two arms. Here in this diagram we see a single chromatid. It has two arms. The two arms meet at a particular point. That is called as primary constriction. What is primary constriction? The two arms of a chromosome meet at primary constriction. The primary constriction has an important structure called as centromere. Centromere is present at the primary constriction. The centromere plays an important role during cell division. Centromere connects the chromosomal arms. During cell division, spindle fibers are connected to the centromere of the chromosome. During cell division, chromosomes move towards opposite pole. This is due to the contraction of spindle fibers. These spindle fibers are connected to the centromere of the chromosomes. So centromeres are important for cell division. If centromeres are damaged, it will affect chromosomal movement during cell division. That will result in abnormalities in the young ones. What is primary constriction? It's a place where centromere is present, and the constrictions other than primary or secondary. This is secondary constriction. This is also another secondary constriction. Spindle fiber is connected to which part of the chromosome? Spindle fibers attached during cell division at the centromere of the chromosome. What are secondary constrictions? The constrictions other than primary or secondary, they may be at any place of the chromosomes. Some of the chromosomes have secondary constriction. It may be more than one also. This is a secondary constriction. Some of the secondary constrictions are called as nuclear zone or nucleolar organizer. We know what is nucleus and nucleolus. Nucleolus is present inside the nucleus. Some of the secondary constrictions carry information for the formation of nucleolus. Such secondary constriction is called as nucleolar organizer. Formation of nucleus inside the nucleus is influenced by this constriction. End of the chromosome is called as telomere. So both the ends have telomere. Telomeres are present at the end. Telomeres provide stability to the chromosome. Chromosomes maintain their length, their structure. This is because of telomere. Yes, a broken piece of any other chromosome cannot attach to the chromosome because telomere prevents the attachment of another chromosome part with this chromosome. So telomere provides stability to the chromosome. As cell divides every time, they become shorter. The telomere becomes shorter at every cell division. One cell divides into 2, then 4, then 8. At every cell division, the telomere becomes short. When the telomere becomes very short, the cell becomes aged. So telomere acts as aging clock in every cell. Satellite is a small part of chromosome, small knob-like chromosome that is connected to the secondary constriction. What are satellites? Satellites are knob-like appendages at one end. Chromosomes containing satellite are called as sat chromosomes. What we see here is sat chromosome because it has satellites. The chromosomes are made up of DNA, RNA, chromosomal proteins. There are two kinds of chromosomal proteins, histone and non-histones. Chromosomes are made up of metallic ions, calcium and magnesium ions. Chromosomes are made up of number 1 DNA, number 2 RNA, number 3 chromosomal proteins like histone and non-histones, number 4 metallic ions. When we see the structure of chromosomes, we have to explain primary constriction, secondary constriction, telomere and satellite. Chromosomes are classified into types based on the position of centromere. Based on the position of centromere, chromosomes are classified into telocentric, acrocentric, submetacentric, metacentric. Telocentric chromosomes have centromere at the end. The word telo refers to the end. And the chromosomes are rod shaped. Acrocentric chromosomes, in acrocentric chromosomes, telomere is found near the end. This chromosome has unequal arms, short arms and long arms. Acrocentric chromosomes are also rod shaped. The word acro also refers to end. Telocentric, the centromere is present at the end. In acrocentric chromosomes, the centromere is present near the end. In submetacentric chromosome, the centromere is present near the center. just away from the center. These chromosomes are J-shaped or L-shaped. Metacentric chromosomes have two equal arms. They have two equal arms. Centromere is present at the center. The word meta means center. These chromosomes are V-shaped. The chromosomal shape, V-shaped, L-shaped, rod-shaped, these shapes can be seen during cell division. During cell division, the chromosomes move towards opposite poles. At that time, we can recognize the shape of the chromosomes. And chromosomes are classified into types based on their function, based on their role. Chromosomes control characters. This is their role. Autosomes and allosomes are the two types of chromosomes based on function. Autosomes control some characters. They control body characters. They control somatic characters. Example, height of the individual, color of the individual, hair nature, eye color. So body characters are controlled by autosomes. Number of autosomes are equal in both male and female. In human being there are 44 autosomes. Allosomes decide whether an individual is male or female. They involve in determining the sex. There are two types of autosomes. They are X and Y chromosomes. The structure of X and Y chromosomes are different. And male and females have different types of different sets of chromosomes. Males have one X and one Y allosome. Female individuals have two X chromosomes. Male individuals have one X and one Y chromosome. Female individuals have two X chromosomes. Based on the role or function, chromosomes are classified into autosomes and allosomes. Karyotype refers to the number, length and shape of the chromosome. Number of the chromosome of a species is constant. We have 46 chromosomes. Chimpanzee there are 48 chromosomes. Chromosomes occur as pairs. 22 pairs are autosomes in human beings. In human beings, the 23rd pair is the allosome or sex chromosome. The number of chromosomes in body cells and gametes are different. Body cells have two sets of chromosomes. In body cells, chromosomes occur in pairs. This condition is called as diploid, 2n. In gametes, single set of chromosomes are present. This condition is called as haploid or n. Body cells have diploid set of chromosomes. Gametes have haploid set of chromosomes. So these chromosomes can be studied by using karyotype. Karyotype shows the number of chromosomes, shape of chromosomes and the position of the centromere. This is normal karyotype. Female individuals have 2 X chromosome in 23rd pair and male X and 1 Y chromosome. During cell division at metaphase, the chromosomes are arranged at the middle. The metaphase chromosomes are isolated and they are arranged in pairs. This is called as karyotype. If we take photograph or have diagram, take diagram, that is ideogram. What we see here is a karyotype. Karyotype is a number, size and shape of chromosomes. Diagrammatic representation of the karyotype or photograph is ideogram. Ideogram is the diagrammatic representation of karyotype of a species. This is normal karyotype. In abnormal karyotype, there may be change in the shape of the chromosome or number of chromosomes. So karyotype is useful in identifying some diseased conditions. So in this part we have discussed about the structure of chromosome, number of chromosomes, structure of chromosome and types of chromosomes and karyotype.